Friends, welcome to Dr. Varsha Gaitonde's channel. In this video, we shall see the different sources of cytoplasmic male sterility or genetic male sterility or cytoplasmic genetic male sterility sources in different field crops. So we'll go one by one. The first crop is maize. In maize, three cytoplasmic sources are available. One is CMSC, that is Charuva, taken from Charuva cytoplasmic source. CMSS from USDA. CMST is Texas cytoplasm. For CMSC, RF4 is the restorer gene. Here, spontaneous reversion is relatively high. CMSS is having restorer gene RF3. Here also the spontaneous reversion is high, and these two are commercially used but not as popular as CMST, which has two complementary gene RF1 and RF2. But CMST is having different problems, like if it is used, it reduces the yield up to 2 to 4%, susceptible to Helminthosporium blight. Next, we'll move to Nicotiana tobacco, that is tobacco plant. This has three cytoplasmic wild sources taken from Nicotiana debnei, Nicotiana megalocephala, and Nicotiana begelovi. These result in poor growth, floral abnormalities, and commercially not used. But these are available in case of tobacco. Okay. Then in case of wheat, Triticum aestivum. Wild sources for CMS are Triticum timophyvi, which has RF1, RF2 or like restorer genes. This is commercially used, require two genes for restoration. Then Aegilops kochai, which has one gene for restoration, and this is commercially usable. Then Aegilops crassa, which has RDF1 and multiple genes as restorer source, which is commercially unstable and photoperiod sensitive. So Triticum timophyvi is the only source which is popularly utilized in case of wheat. When we talk about cotton, Gossypium hirsutum, tetraploid cotton, here three cytoplasmic sources are there. One is Gossypium annamalum, Gossypium arboreum, and third is Gossypium herkensi. Herkensi is commercially used, but it leads to 50% reduction in yield, whereas annamalum and arboreum are not used commercially. Sunflower is Helianthus annus botanically, which has PET1 as cytoplasmic source. This has two polymeric gene as restorers, RF1 and RF2, commercially used. In case of onion, again we will see the three cytoplasmic sources, CMS, ST, and Nasic white globe. Among the three, Nasic white globe is commercially used, and for this restorer genes are known. In CMST, two independent systems, that is gene A and gene B and C, which show complementary action. These are thermomobile, having 93% sterile plants at 14 degrees Celsius and 10% at 23 degrees Celsius, which are commercially used. Next is Bajra, Penicetum glaucum, species monody. Here the CMS source is Tefton A, which is commercially usable. Then Ludhiana A2, but A4, AGP, and A5 are most stable but less commercially used as comparing to Tifton A. Then when we talk about rapeseed and mustard, first species is Brassica napus. This has cytoplasmic source CMS NAP, which is controlled by 1 to 4 restorer genes, unstable at high temperature. Then CMS ogu, that is introduced from wild radish. This is susceptible to low temperature, then chlorosis, lack of metabolic activity, then commercially unstable, but ogu is certainly used. Pole has restorer gene as RF1, RF2, which is taken from Brassica napus. This is most stable. Restoration is affected by modifier genes, is the only problem here. Then CMS TOR is taken from TON40, which has RF gene in Brassica napus. Restoration is complete in TOR cytoplasm. Next species is Brassica campestris. Here CMS MUR, which is taken from Diplotaxis moralis. Here the restorer gene occur in Campestris, taken from Campestris, which is used commercially. Next is Brassica juncea. Here the cytoplasmic sources are CMS ogu, TOR, MS4 and juncea. Again, two wild forms are there, Diplotaxis barthotti and Moracantia arvensis. CMS ogu has where RF gene is available. Here, defects arrived using somatic hybridization, means defects are removed. Then in CMS TORN40, RF gene from Brassica juncea are obtained, which are highly stable, but incomplete restoration is the problem. In case of CMS MS4, this has single dominant gene as restorer. Here, perfect maintainer is not available. In case of CMS juncea, restorers are available and commercially utilized. Diplotaxis barthotti has one gene as restorer form, but it shows gametophytic action. Then Mercantia arvensis also has one gene in restorer form. This is also having gametophytic action. Then red gram or tool, Kajan is the scientific name. This has A4 and A2 cytoplasm. A4 is the alloplasm taken from Kajanifolius. Restorers are available here. Hybrid ICPH 2671 and other hybrids are commercially produced by using this source. A2 is taken from Kajanus scaraboides, which has where restorers are available, and GTH1 hybrid is released by using this particular source. If we talk about sorghum, here different cytoplasmic sources are available like Milo, which is A1 taken from bicolor. Restorer gene is taken from Kafir race, that is MSC. Milo is the only CMS which is commercialized. Then again A2 is available, with where restorer gene is taken from Guinea, A3 from Caudatum, A4 Guinea, then A5 from bicolor, and A6 is Dura. A2 to A6 are not used commercially. Then rice, in rice different species are there. For different species different CMS sources are taken. Oryza spontanea has CMS CW, which is a Chinese strain. Then CMS WA is a wild abortive strain. Then these two are widely commercialized. Then in case of Oryza sativa, CMS BO, that is taken from Chinsuraka boro 2, which has single dominant and gametophytic control, stable one. CMS ARC, which is taken from ARC 13728, having 16 strain, but it is unstable. Oryza perennis, CMS perennis is the source which is usable. Then Oryza rufipagon has CMS W18, which is taken from W1080 strain. Friends, the information furnished by me, I hope that is helpful to you. If you liked this video, please do not forget to like, keep on commenting, sharing, subscribing my channel. Thank you once again.